Welcome to another tutor short provided by the Educational Support Services Department of Lehigh Carbon Community College in Snexville, Pennsylvania, which is just outside of Allentown. These videos review key learnings for the science courses provided here at L-Tri-C. And please remember that the Educational Support Services Department does provide walk-in tutoring 5 days a week.
Hello and welcome to the Anatomy and Physiology 2 tutor short. On the blood, we'll be discussing about the differences between erythrocytes and leukocytes. Erythrocytes, also known as red blood cells, are biconcave, anucleate disc, which means they don't have a nucleus. They are salmon colored and have a diameter of about 7 to 8 nanometers. Their function is to carry oxygen to the body tissues and carry away some of the carbon dioxide. Their duration of development is about 15 days, and their typical lifespan is about 100 to 120 days. To the right, we have a picture of an animated version of a red blood cell. You can see how it is biconcave and disc-like right here. The bottom picture shows a red blood smear using a Wright stain. This is what they typically look like underneath the microscope. We can divide the leukocytes into granulocytes and agranulocytes. The first three leukocytes we will be talking about are granulocytes. So granulocytes include neutrophils, basophils, and eosinophils, and have a roughly spherical shape. They are larger and have a much shorter lifespan than erythrocytes. All granulocytes will have a lobed nuclei, and their membrane-bound cytoplasmic granules stain quite significantly with Wright's stain. Functionally, all granulocytes are phagocytes to a greater or lesser degree. On this slide, we will be talking about neutrophils. Neutrophils have a multi-lobe nucleus and account for 50 to 70% of the white blood cell population. Neutrophils are about twice as large as erythrocytes. Neutrophils contain very fine granules. The reason why they are called neutrophils is because they are neutral-loving, because their granules take up both basic and acidic dyes. Together, these dyes will make this lilac color, as you can see through here in the cytoplasm. The duration of development for neutrophils is about 14 days, and their lifespan is about 6 hours to a few days. Their main function is to phagocytize bacteria. As you can see through the pictures on the right, they can range anywhere from 2 up to 6 lobes per cell. The next granulocyte is the eosinophil. They only account for about 2 to 4% of all leukocytes and are approximately the same size as a neutrophil. It is a bilobed nucleus cell and has a red cytoplasmic granules, which you can see through here, all the red granules in the cytoplasm. These granules are lysosome-like and filled with a unique variety of digestive enzymes. The function of eosinophils is to kill parasitic worms and also play a complex role in allergy and asthma. On these slides, you can see the variations of an eosinophil look like. Some could be a little bit spread apart, they could be connected, but essentially they all have 2 lobes and a very dark red granulated cytoplasm. The next granulocyte is the basophil. Some people like to call this the big bad basophil because it's almost twice as large. Basophils are the rarest white blood cells and only average to only about 0.5 to 1% of the leukocyte population. Their cytoplasm contains large coarse histamine-containing granules and have affinity for the basic dyes, so basophil, base-loving, and the stain is purplish-black. The histamine is an inflammatory chemical that acts as a vasodilator, which makes the blood vessels dilate and attracts other white blood cells to the inflamed site. The function for this is to release the histamines and other mediators of inflammation, which also contain heparin, which is an anticoagulant. Here on the right, we can see the different variations underneath the microscope. You can see sometimes you can't even see the nucleus because it's so heavily granulated. So if you cannot see like the cytoplasm and it is very dark and huge, you probably are looking at a basophil. So our agranulocytes consist of lymphocytes and monocytes. The lymphocyte has a spherical nucleus or sometimes indented nucleus and has a pale blue cytoplasm. Its diameter is about 5 to 7 nanometers, so it's about the same size as a red blood cell. Its duration of development is up to days and weeks and can last for either hours or years. Its main function is to mount the immune response by direct cell attack or via antibodies. They only account for 25% of the white blood cell population, and they are the second most common leukocyte in the blood. When stained, a typical lymphocyte will have a large dark purple nucleus that occupies most of the cell volume. Sometimes you can even see a halo of the cytoplasm around the dark nucleus, as you can see in this photo right here. Sometimes you won't even see the cytoplasm, you'll just see a dark mass, but you won't see any granulation. The function is also to produce antibodies, release chemicals onto various infected cells, which will kill them. And the second agranulocyte is a monocyte. Typically their nucleus is either in a kidney or U-shaped. They'll have a gray to blue cytoplasm. Here is a U-shape, here is more of a kidney shape, and this one doesn't even have that shape. Monocytes account for 3 to 8% of white blood cell population. They are the largest leukocyte. When circulating monocytes leave the bloodstream and enter tissues, they will differentiate into highly mobile macrophages. Thank you for watching. To view any of the models, or if you have any questions, please come to the Science Lab in SH 150 in the Educational Support Services Center. Thank you.